This is Biology 1, Chapter 11, Part 3 on complex inheritance and human heredity. In this part of the video lecture, we'll be taking a look at chromosomes and heredity, and specifically taking a look at what we call karyotypes and looking at some chromosomal disorders. A karyotype is what the medical field uses as like a picture of a person's chromosomes. Karyotypes are taken when a cell is in metaphase. So they freeze the cell using a special solution called colchicine, and they freeze those chromosomes right in the middle or the equator of the cell. And then now what they do is they take them digitally and they arrange them in size. Remember, humans have 23 pairs of chromosomes. We have 22 autosomes or body chromosomes, and then there's the one pair of sex chromosomes. So when taking a look at a karyotype, it is very easy to tell the difference between a male and a female. If you look after chromosome pair number 22, you will see a spaces for X and Y chromosomes. The color karyotype on the left is the male karyotype because there's one X and one Y chromosome, and the female is the black and white copy that is on the right side, and you know that that's female because there's two copies of an X chromosome there. So chromosomes can, once they're in a karyotype, they can show abnormalities. So karyotypes can be looked at a person in the medical field, making sure that there are the correct number of 23. Are they all the correct size? Karyotypes are only done in situations where there more than likely is some type of chromosomal disorder, and they need to find out specifically what chromosome or where it is. We talked about in our last chapter how cells have a life limit, and there is a certain amount of times that a cell will divide, and then after that the cell will undergo apoptosis or programmed cell death. So how does the cell know when it's supposed to die? How does apoptosis start in a cell? Well, cells have on the ends of the chromosomes, kind of like the little shoelace protectors that are on the end of your shoelaces. Those keep your shoelaces from fraying and becoming difficult to tie, and telomeres are little protective caps of DNA and proteins that are on the end of chromosomes. And every time the cell divides, a little piece of that telomere is lost with every division. So telomeres start out very long, and they get smaller and smaller as the cell keeps dividing. When the telomere is gone, then that is when apoptosis is initiated and the cell will die. There is research going into telomeres as far as kind of the idea of anti-aging. The only reason why our bodies get old and we eventually die is because all of our cells get old and they eventually die. Theoretically, if you could keep the telomere piece on the ends of the chromosomes longer, you could extend the life of a particular cell. And if you can do it with one cell, you should be able to do it with all of the cells in your body. But the only side effect of some of these different types of treatments that they are trying to provide for telomere research is that they can also cause cancer, because what ends up with happening with cancer is that the cell believes it still has its telomere on the end of its chromosomes, and it does not, and so it keeps dividing again. That's why cancer is our uncontrolled and unrestrained growth of cells. Non-disjunction is a condition that can happen during meiosis. Remember, the cell cycle has to go 100% correctly 100% of the time. If meiosis runs properly, every cell should have the correct number of chromosomes. So they should have 23 chromosomes in sperm and 23 chromosomes in egg. What ends up happening sometimes is that those chromosomes actually fail to separate during meiosis. And so during anaphase 2, when we have the splitting occurring, they're not split in an even sort of way. So some cells will end up with extra copies of chromosomes, and some cells will have not enough chromosomes. When we have an extra copy of the chromosome, we call that condition trisomy, because tri for 3. Monosomy is when there is only one copy of a chromosome in a cell. Now, this can happen from either abnormal sperm that fertilize a normal egg, or an abnormal egg that's fertilized by a normal sperm. But here we have in the diagram right here, you can see the monosomy that ends up happening because the new cell, the diploid, which should have a full 46 chromosomes, only has 45. You can't split up 45 in an even way. So some cells are going to have some extra chromosomes, some not enough. Down syndrome is a common name for a condition called trisomy 21, when an extra copy of chromosome 21 is inherited. There can be many mental and physical issues with Down syndrome. There's, again, a range kind of from mild to moderate to severe, but in general, with some aid and facilitation, these folks can live a fairly normal life. Non-disjunction can also happen with sex chromosomes. Remember, normal males and females, normal female, XX, normal male, XY. But in the case of during meiosis, if those chromosomes do not split out correctly, you can actually get different combinations that can produce different disorders. A female, for example, that only receives one X chromosome, and that's it, would be a female with what's called Turner's syndrome. Turner's syndrome females tend to have underdeveloped reproductive organs. It would be very difficult for a female with Turner's syndrome to become pregnant. Ironically enough, if a female gets an extra X chromosome, there is not a whole lot of differences developmentally, and it's not like it would be a super female that she would have an extra X chromosome, so she would be like the ultimate female. That's not the case. An extra X chromosome doesn't seem to make much of a difference for females. Normal male, again, XY. If a male receives an extra X chromosome, then he will be a male with Klinefelter's syndrome. Again, Klinefelter's males would have underdeveloped reproductive organs. It would be very hard for a Klinefelter's male to have biological children of his own. If males receive an extra Y chromosome, it does not make them supermen or super males either. There's very little effect. Everyone needs at least one X chromosome. If for some reason during fertilization the developing egg only receives a Y chromosome, that's not enough information to keep going for the cell, so that will result in death. There are different tests that can be performed before birth to detect certain genetic disorders. These would only be done if there was, for example, a family history of certain types of conditions or disorders that would be of concern to the parents. Amniocentesis is a process where they take a very long needle and they withdraw some of the amniotic fluid from inside the uterus, the placenta rather, of the female. The amniotic fluid has old cells that have been sloughed off from the fetus, and so they can take those cells and screen them, make karyotypes out of them. Chorionic villus sampling is where they place a catheter up the cervix of a female, and they kind of take a little scraping from what's called the chorion, which is right outside the amnion, and there's a lot of really blood-filled, chromosome-rich sort of area. So again, they could do a karyotype from that as well. In the cases of perhaps wanting to know if an offspring was in hemophiliac, they can do fetal blood testing. They take a rather large, again, needle and place it up the cervix, and they take a very small sample of blood from the top of the head of the baby to collect for analysis, again, to see blood type and to see what, if any, blood disorders there may be present. So this has been Chapter 11, Part 3 on complex inheritance and human heredity.